There'll be a one for Wednesday. Hopefully it doesn't take as long for the Wednesday one to show up as the Monday one to show up. Hopefully it should be pretty quick. But without those questions, I don't know if anybody has any questions about Monday's lecture. I want to sort of voice today. Or we sort of continue our talk on repair mechanisms. What's on the board is a summary of at least these top two things. The bottom one is a completely different type of system we have to deal with. But these two are related in a fashion. And they both sort of do the same thing. There is a damaged nucleotide that is the issue. Remember, with pyrimidine dimers, we have two nucleotides that are connected together, so it's not a single nucleotide that's damaged, it's sort of two together. It's called multiple in parentheses. There are other types of damage, but these are the ones we're sort of going to deal with as the more common types of things. And the repair mechanism that does these things is something different. So a different set of enzymes, a different mechanism. The thing that does this, a different set of enzymes, a different mechanism. The thing that does this, a different set of enzymes, a different mechanism. So in total, it's going to be a lot of proteins to memorize and where they work and what they do, and all of the fun stuff. Yeah. This is even the hard part of exam 3. Yay! Okay, fine. Dive right in. The other thing that gets confusing with students, I know I said dive right in, but the other thing that gets confusing with students is the similarity in what the first two things are called. Nucleotide repair versus nucleotide excision repair. Easy to get these two things confused because the naming system is basically the same. So try not to get confused. We have a way to repair a single nucleotide and a way to repair a stretch of DNA. So if you want to repair some of the things that are causing single problems, get the depurination, the lack of a base being present, deamination, alkylation where a base has been modified in some way, or the incorporation of a base analog was not caught during replication. These are things that can be fixed later on. Depurination, deamination, alkylation are things that can happen anytime, but base analog incorporation does require replication to work. So it is kind of a different type of thing to think about than the other three, because it only affects things when you are copying your DNA. The other things will physically damage a nucleotide. So you'll want to put this one in a little bit different category than the other ones, because of sort of when it occurs. Chances are great that this will not be an important thing to memorize for a quiz or exam, but it is sort of a way that we can distinguish some of these things as we go forward, at least mentally speaking. To do a single nucleotide removal, we need two basic types of enzymes. One enzyme that will remove the base, and then a second enzyme that will remove the sugar from the phosphodiester backbone of the DNA molecule. Why do we need two things? Why can't we simply remove the nucleotide whole as it is? Does anybody have any thoughts? And as you think about it, look at our board and think about how one of these is different than the others. And exclude number 4, just look at number 1, 2, and 3. Remember anything about what depurination does, deamination, and alkylation do? If you're paying attention, I kind of gave the answer away. What is the difference between those first 3 things? 2 of them do the same thing or a similar thing. The third one does something different. Remember thoughts? And I really just mentioned it a second ago. When we think about deamination, we want to remember that we are modifying the base by removing an amino group from the purine part or the pyrimidine part of the nucleotide. And when we alkylate, we are also modifying the base. When we look at depurination, the base is physically removed, leaving the sugar phosphate backbone intact. So the fact that this one leaves half of the nucleotide present sort of explains why we need a 2-step process. Because one of these only needs one step. Only one thing needs to be done. or the other one, sort of two things need to occur. Which is why I sort of can explain why we need both of these, because we have two different types of damage going on. One where we change the base, the other where the base is gone completely, which does necessitate two different types of approaches, which are sort of linked together. And so the first one is what we call a DNA glycosylase. This is an enzyme or a group of enzymes that will remove a base from a nucleotide, leaving the sugar phosphate backbone intact. The last thing is called the AP endonuclease. Doesn't matter what A and P stand for, but we should definitely know what nuclease means. Anybody? Nuclease? What does nuclease do? I'm gonna go with a more general answer, which is cuts DNA. Nuclease is cut DNA, but breaking down is sort of the same thing. Just I think about it as cutting more than degradation, but it's still together. And endo tells us where the cutting is occurring. So where is an endonuclease cut compared to an exonuclease? An endonuclease is in the middle, exonuclease is on the end, and sort of choose backwards. Remember, proofreading activity, exonuclease activity, moving backwards, removing the base, continuing forward, versus cutting the DNA strand at a particular location. So this cuts the nucleotide in half, this cuts the DNA. So as we look at the depurination reaction, the base is already gone, so we don't need the DNA glycosylase. Its work has already been accomplished. So the only thing that we need for depurination is the AP endonuclease. I got my slides mixed up. Wait, no, it is depurination. I misread that, sorry. Depurination creates an apurinic site. The base is gone. We don't need DNA glycosylase, but we do need the second thing, which is the AP endonuclease. Because the sugar phosphate backbone, the sugar's still there, we need to get that out so we can put in a new nucleotide in its position. And so what you need to imagine, sort of what picture are we trying to think about here? It should be fairly straightforward. If we sort of imagine a piece of DNA, to make it really easy. If something happens where, like depurination, we're going to remove that base, we still have our sugar phosphate backbone intact, and there's nothing opposite that T. So what we need to do is to make it so that we can add in a new nucleotide to base pair with the T. So our AP endonuclease is going to cut two places on either side of the phosphorus bond of the missing nucleotide. And once that happens, we have a gap. And now we have something that just looks like replication. We've got a primer, which is a piece of DNA that can be added onto, and DNA polymerases will come in and add a nucleotide. So basically, in almost all repair mechanisms, the last two things that happens is DNA polymerase fills in the missing area, and DNA ligase connects everything together, which is the last two steps of replication. We're kind of using a similar mechanism that we already do. Remove some DNA, and then fill in some DNA. Like replication, it was actually an RNA primer that was removed, and then filled in with DNA, and then glued back together as the last steps of replication. But the AP endonuclease, because it's an endonuclease, cuts somewhere in the middle, not in the end, but it also cuts twice. And it's only cutting one strand. It's not affecting the other strand whatsoever. Only one thing is wrong, so only cut one thing. Now what you should hopefully recognize or appreciate that this is a similar thing that we talked about already, mute H, or mismatch repair is doing the exact same thing. But there it was cutting really far away, so we remove a whole bunch of DNA. Here there's only one thing being removed. So it's a much more surgical strike. And what allows it to be surgical is that these nucleotides are modified or changed in dramatic ways that can be easily noticed. It's hard to notice if a right nucleotide is on the wrong place. It's very easy to notice that a wrong thing is in the right place. So only one step is needed to deal with the depurination step, or what we call the apurinic sites. Well, for deamination, or deaminated nucleotides, alkylated nucleotides, or even the incorporation of base analogs, we need both steps. And the first step is going to be the DNA glycosylase, removing the base, which will create either an apurinic site or what we call an apyrimidinic site. A nucleotide that's lost its base. Depurination only affects A's and G's. The DNA glycosylase can work on anything, so C's and T's also get removed, which is why we get this apyrimidinic site. This simply means the base is gone, the sugar phosphate backbone is still intact. So the first step is remove the damaged nucleotide of the base, and then the second step is remove the rest of the nucleotide. So one whole new one can be slotted into that missing location. And so really one of the harder parts about this is simply remembering that one requires two things, the other only requires one. So this is just second step only. This is first and second equal. And this is true for all of those three. It's a fairly simple process. You can cut DNA, remove DNA, replace DNA. Has anyone ever tried to patch a piece of clothing? Typically, you don't just put a patch on top, you remove some of the damaged area and then you put something on it. That's kind of the same thing. Even if we're talking about medicine, just put a Band-Aid on top of something, you might clean the wound, remove any damaged skin or something around it, and then fix it. That's what we're doing. Cutting something out and then putting in something new. But it's only one nucleotide being removed, which is the important thing about this process versus the next process. It's not going to be important for exam 3 because proofreading activity is on exam 2. For the final, you might want to start thinking about proofreading activity is doing a similar thing to this nucleotide repair system. One nucleotide removed at a time and fixing it. It's just proofreading is only at the end when we're breaking DNA. These things can happen anytime. Any general questions about which enzyme cuts the DNA and which enzyme removes the base? Because again, DNA cutting enzymes will be important to remember as we go forward. So this is kind of like proofreading activity, and our next mechanism is very similar to the mechanism of mismatch repair. Mismatch repair, there's no bad nucleotide, just an incorrect nucleotide. And we rely on methylation states to identify the original piece, which is the right nucleotide, and the new piece, which is the wrong nucleotide. For example, nucleotide excision repair is used to fix thymine dimers or other pyrimidine dimers quite a lot. So we typically associate this with fixing UV damage, whereas these are fixing other types of damage. Basically, most of these are caused by exposure to different types of chemicals. This is something that is from radiation. Now, a pyrimidine dimer is something that is fairly unique, and it really creates this bulge in the DNA that kind of looks like this. And this is a noticeable structure. It's a change in the DNA topology that can be identified by repair mechanisms. So what happens is there's this repair mechanism sort of going around looking for damage, and it spots it. So that's wrong. Let's remove it. And instead of removing just the two nucleotides that are wrong, it says nucleotide, and cut a lot of DNA out and just fill it all in. Which is a lot like mismatch repair. We don't remove one, we remove a lot, and just slap on some new stuff because that's more efficient. But just like the AP endonuclease, the enzymes that are involved in this process are cutting a single strand in two different locations. So we can excise the damaged piece of DNA. Once that damaged piece is removed, new undamaged DNA can be put in its place. A large chunk of DNA being removed versus one nucleotide being removed. Generally speaking, we call the enzymes that do this NER, endonucleases, which should be pretty easy to remember, because it's nucleotide excision repair. But unfortunately for you, we do want to talk about some specific examples. So we have, for bacteria, the enzymes involved in fixing UV radiation damage are called UVR. There's a few of them, one's A, one's B, one's C, you can memorize that particular part of it. But UVR should be pretty easy to think about. UV is UV, R is for radiation, or it's for repair, or it's for both. Hopefully we can make that connection pretty easily. This also illustrates one of the things we have to deal with in science is the naming convention in bacteria and prokaryotic organisms versus the naming convention used in eukaryotic organisms. If you get into this field, not that you need to memorize this for a test or exam, but if you look at things, these always mean bacteria, and if it's written like this, it always means eukaryotic. Because that's just the way it is. So what do you think RAD might stand for? Besides cool. Vivacious. Toby, do you do there? Radiation. Yeah. Well, or maybe not. But radiation is a good guess. Because we're going to see rad proteins be involved in something that's not caused by radiation. Wait, no, it is caused by radiation. That's going to be okay. But AD endonuclease, NER endonucleases do exactly the same thing. It's just the size of the DNA removed is different. It's not DNA glycosylase and AP endonuclease, it's either UVR proteins or a group of RAD proteins for UV damage repair.
So the red does stand for radiation.
It's not important.
Okay.
Memorize that fact. But some people talk about repair as the R, and I forget what the A and D sometimes stand for. But there are a bunch of proteins that deal with radiation damage in eukaryotic organisms, and they're numbered. We will memorize a number in a second. You don't need to memorize all the numbers involved in this process. Just like I don't care that you remember what UVR stands for, it's not an acronym that's going to be easy to explain or define on an exam or quiz. Just UVR proteins go with nucleotide excision repair, which fixes UV damage, which is pretty daggers. Make sure we can link those things together. In humans and other animals, we use RAD proteins to do the same thing in a similar mechanism. And every once in a while I like to include papers, or at least some abstracts of papers, to whet your appetite for getting excited about science. And to show you that these things are not just boring, stupid shit that we learn in class. It could actually help the real world. So if you wanted to study things like this, probably not chromatin compartmentalization and how it regulates response to DNA damage. But maybe you care. Maybe you care how DNA damage is fixed in a world where all your DNA is wrapped around proteins already. For people that study DNA, that's a pretty important thing to think about. How does chromatin affect what other proteins are doing? Especially important for replication. How does DNA polymerase get access to the DNA if the DNA is already bound by histone proteins? How does transcription happen if the DNAs are already surrounded by histone proteins? So there's a lot of stuff we're not talking about in this class that impacts a lot of things, but a little bit deeper than we actually care about. These are very easy to fix and typically very fast fixes. And typically no changes are introduced when we do nucleotide excision repair or the nucleotide repair system. Unfortunately, there is a much harder type of damage to fix, and that is what we call a double-stranded DNA break. Technically, what we have on the board behind us is an example of a single-stranded DNA break, and that's not exactly the thing. Well, when we talk about a break, we're talking about a single phosphoester bond being gone. So if we look at this as an example of a starting place, which is some new picture we're looking at, a single-stranded DNA break would just give us this, where nothing else is happening, but we are breaking one phosphoester bond. We don't talk about fixing single-stranded DNA breaks at all, because they are very easy to fix. Because this just requires DNA ligase, just like anything else we would do. So very easy to fix, single-stranded DNA break. And nothing really bad happens here, because everything's still connected together very easily. But if we have a double-stranded DNA break, that is when this type of picture would start to show up. Where now we're cutting both strands. No nucleotides are gone just yet, but we are physically separating one side of the DNA from the other side of the DNA. And as we talked about with telomeres, when we cut the DNA, it allows it to sort of lose its cohesion, which you call the breathing process. And when DNA breathes, DNA gets digested. So we start to lose DNA fairly quickly when there's a double-stranded DNA break. So you can lose lots of DNA very quickly if you're not fixing these fast enough. This is why double-stranded DNA breaks are such a big problem, is you immediately start losing DNA. Now luckily there's a way that we can get this back to normal-ish fairly easily, and there's a way that we can have a band-aid on it and hope for the best. Those are the two sort of ways that we can deal with things. Now the reason why the cell does this is because single-stranded DNA is considered to be viral or foreign DNA, which we want to try to keep from causing us damage. So we typically deal with things that are foreign as things to destroy. That's what our immune system does. And our cells have their own immune system inside of them we don't talk a whole lot about. The methylation system in bacteria is also an immune system thing. Any DNA that's not methylated gets nucleated or digested because it's thought to be foreign and wrong. But here we're on the end, so it's kind of exonuclease activity. What you have to remember is the exonuclease activity goes both directions. And so it's not just treating one way, it's treating both ways. So our picture gets bigger and our hole gets bigger and bigger because we're chipping away at both ends as we go along. Now luckily for us, there is something cool that happens that allows the rest of the thing to happen, and that is the digestion that we see happening is an uneven digestion. And it leaves what we call a 3' overhang. So this little bit hanging out here is the 3' end of the DNA molecule. And I know that because that's what this thing does. It leaves extra nucleotides on the 3' end that don't have anything to base pair to. Sort of like a little fringe hanging out there. So I don't know why it does this, but for whatever reason, the nuclease activity is different on one side versus the other side, or sort of top versus bottom. But we need that 3' overhang to do the repair mechanism that is going to be best for us. UV light causes thymine dimers and cytosine dimers. What causes double-stranded DNA breaks is most often ionizing radiation. So exposure to gamma rays and X-rays are the two most common things that are going to cause double-stranded DNA breaks. As you can see, there are other things. So anytime you move DNA molecules, like for transposons or do other things, there are ways that we can cause double-stranded DNA breaks other than this. But if I ever ask you, and I will ask you this, what is the cause of a double-stranded DNA break? This is the only answer: ionizing radiation. And when we fix ionizing radiation, we do one of two things. We call recombination repair, or what's called non-homologous end joining. Recombination repair is the more complex, but the better mechanism. And non-homologous end joining is quick and simple, but not so good. Now, what I want you to sort of keep in mind as we go forward Recombination repair is basically recopying everything. You may make a few mistakes, but at least you're getting everything back. Whereas non-homologous end joining, if you like ripped a bunch of papers out of the book, you just put the book back together and say, okay, here's what's left over. So you're missing a lot of things, at least you have a whole book. Or at least the book is still back together as a book. When we talked about crossing over, which was the thing that happens during prophase 1, where DNA from one homolog is swapped with DNA in the other homolog. So we're gonna think about that in the same way here. We're gonna use something, we need something to use as a template or a pattern to replace something we miss or have lost. So recombination repair does require a copy to be present. Sometimes you don't have a copy, which is why you have to do the other thing. We do need something to use to fill in the missing pieces. And for diploid organisms like us, there's always a homologous chromosome that's present. So we always have something that should have the same gene sequence and the same gene order, same gene content, not necessarily the same gene sequence. Remember, some of us can be heterozygous, have a dominant allele in one chromosome and a recessive allele in the other chromosome. Even if we're homozygous, the DNA sequence might be different because of what we call single nucleotide polymorphisms. It's never going to go exactly back to 100% the same, it's going to be close. For bacteria, sometimes this can occur, and what we don't talk about in bacteria is most oftentimes they have multiple copies of their chromosome to prepare for cell division. So it's always like an extra piece sort of hanging around that you can use. It's not a diploid state, but there's always an extra copy there. So bacteria do this quite a bit, they're really good at it. Eukaryotic cells do it, they're pretty good at it. We need a copy. We use the copy to replace a missing piece of DNA. And because our copy may not be exactly the same, sometimes we'll get something that results called gene conversion. We'll talk about it as we go forward, but not really right now. But it is, things may not be the same at the end as they were at the beginning. That's an easy way to think about gene conversion. Something gets changed. And as you can see on the last line, recombination repair can occur with and without crossing over. Does this confuse anybody? Didn't we talk about recombination equaling crossing over? Like they're the same, like synonymous terms, kind of? Did we not talk about that? So it'll be weird. We'll explain it. Hopefully it'll make sense what we actually mean when crossing over happens versus what recombination repair is. Because remember, with crossing over, a double-stranded piece of DNA from one chromosome has to switch places with a double-stranded piece of DNA in another chromosome. That's what crossing over means. Some piece of DNA gets moved to the position and vice versa. First thing is some general steps, then we'll look at some specifics and some pictures and some proteins involved in this process. But our picture is going to be important to start with. That 3' overhang is the thing that is starting this process. So when I say free end, that actually means the 3' overhang. So in your notes, you have these slides in front of you. By free end, maybe put a notation, 3' overhang means free end. It'll be in the next slide, but it's not a bad idea to have it sort of here to start with. There are specific proteins that initiate this process and help this 3' end facilitate its movement. So we have proteins binding to the DNA, we have what's called an invasion step. That invasion step leads to a displacement step. Once we invade and displace, we can now do an extension step, which means DNA gets longer. Once the DNA is long enough, we have an injection phase, and then we have a finish phase. I know it says the other things, but so we have invade, displace, extend, eject, and finish. The finish is several complicated things going on. Well, not complicated, but several things going on in this process. So if you keep this in mind, it'll help us understand as we go along the specific things that we're doing here. Before we start, are there any general questions? You want to pray? Do a short prayer before we start? Nobody's religious enough to pray before we start something complicated. Okay then. Recombination repair without crossing over. So we have the proteins that are going to be involved in initiating the recombination repair are called REC-A, or recombination protein A. I want you to memorize that. That's what it stands for, recombination protein A. And then RAD51. RAD51 is not involved in UV light repair, but is involved here in recombination repair. But again, RAD is a eukaryotic protein and REC-A is a bacterial protein, although there are homologs to this REC-A and the archaea. But we talk a lot about bacteria instead of just all prokaryotic organisms. And what these proteins do is they bind to the 3' end of the DNA that has encountered a double-stranded DNA break. And so if you could notice, there are 2 double-stranded pieces of DNA, the red one and the blue one. And the red piece has 2 strands, a light red and a dark red. Just so you can see the 2 different anti-parallel strands of the DNA molecule. And the arrows, if you notice, hopefully you can see they're going in opposite directions, which is important. And DNA interacts in an anti-parallel fashion. Also, you keep in mind that DNA to DNA, RNA to RNA, or DNA to RNA are always anti-parallel and complementary. And so here we have, again, one anti-parallel strand versus the other on both sides. In the cell, up and down are not really what goes on. Sometimes they're right next to each other, left and right. So don't get too hung up on the perspective, this 2-dimensional perspective. There are 2 3' ends. One here and one here. Either one of these can be used for the invasion step. Based on the picture, it makes sense this one goes in because it's closer, but it's not an up-down thing, it's that really goes together, so any one of them can work. But as we go forward, it's this bottom piece here that's going to be starting this process. So we're going to see this be involved in the recombination repair process. And without these 3' overhangs, it doesn't work. What you need to think about as we go along is how does this look like replication? You should see some similarities between the replication process and this recombination repair process. And then down here, side note, is just some other proteins that we'll talk about tangentially as we go forward for the rest of the semester. BCRA1 and BCRA2, which are common proteins involved in repair. But with certain mutations, these can lead to breast cancer, both in men and in women. So typically we see a lot of repair proteins being broken. Cancer sometimes becomes a fairly high chance of happening. So RAD51 and REC-A will bind to that 3' end and facilitate the invasion process. But you can see the blue strand is now being pulled in and is now base pairing in an anti-parallel and complementary way. Ignore the brown part, because that's going on to the next step. We just want to think about the blue part, invade. And so right here, this base pairing is occurring between the template strand that's unbroken and the broken DNA. And you can see the light red one is now displaced. So again, invasion leads to displacement. Trying to slide something in there is going to push something else away, which is what we're doing. Now because this is no crossing over, this pink piece is not going to do anything else. This is going to be bumped away and sort of sit there and wait. And all the magic is going to happen with this little blue piece and what we're going to do with it. We call this structure the heteroduplex because it's made with 2 different DNA molecules. 2 different double-stranded DNA molecules have now sort of entwined in a weird way. And hetero means different, right? And duplex means two, so two different things together. Heteroduplex formation only occurs when we have no crossing over happening. But once we have this base pairing here, this blue piece can act as a primer. And we can simply extend the DNA a certain length. And I don't know how we control how long it is, but eventually we're gonna keep extending that blue piece using the red piece until it gets really long enough. And long enough is a hard thing to define.
You may have said this, but what's the DS for DS?
Sorry, double stranded. DS, double stranded. Like SS stands for single stranded. Sorry, I thought I defined that earlier. Or I thought it was not needed. It's a good to think about because in my role, that's a common abbreviation that not every role. So sorry about that. And so, recombination repair proteins bind to that 3' end, pull it in to the template, pushes the other thing away, so we can use that broken piece as a primer to add on new DNA using this template. We just keep going this way. And again, we want to get long enough, because as we get long enough, this thing, we want to try to make sure that we can bridge the gap. That's the whole point here. Build this thing up so we can get back here and bridge the gap. So as we get this thing longer, it gets long enough. So after extension, we have this ejection process happen, where the new strand gets long enough, the original piece, the template, goes back together, which is an important part of the no crossing over. This piece of DNA has been unchanged. The only thing we do is fix the broken piece. So again, we evade, displace, extend, and then eject. And then it's gonna go back together here in a second. But when we put it back together, we're just really doing a lot of things we already talked about, which is just filling in gaps. And filling in the gaps is a very easy thing for cells to do. But as you can see at the end, at the bottom, it's not all blue anymore. This brown part, at least this brown part, or at least this brown part up to here, or maybe up to here, is actually pink. Because that's what it looks like, because we use the dark red to make this brown. So this little chunk of DNA right here is going to be different than what was there originally. That's why we're not going back 100% the same. There is the potential that whatever we use is not the exact DNA sequence that we need. It's going to be the same genes. We're not losing gene content. Our DNA sequence is maybe changing slightly, or maybe changing more than slightly. The other thing that may happen here is there might be some mispairing that goes on. Because maybe you put an A, maybe there's an A here when there should have been a T here, and now you have a mismatch. So there could be things that you fix after this fact. Again, proteins bind to the 3' end, stick it in there during the invasion process, the base pair to the template, which displaces the original partner. It's like cutting in, so you never try to cut in during a dance. One dance for me, and you can go back to your boyfriend or date. And one shoulder. You're out. Go back and say, no. Mine. My dance. Anyways, no crossing over because the template strand has not been changed. And red stays with red and blue stays with blue. No switching going on there. So everybody leaves with who they brought. Which is a good thing to do at dance. You've never gone to a dance and left somebody else? You're a bad date. Maybe your date was a bad date. You never know. But recombination repair can happen using crossing over. And instead of forming what we call a heteroduplex structure, we form a complicated structure called a holiday junction. Why do you think the word holiday is capitalized?
Somebody's name.
Probably somebody's name. Yay. there was a big sort of, I won't say big, there was a push. I say push not really the right word, but there was a impetus in the scientific community to try to change a lot of these things. So like be descriptive rather than personal. So it'll be easier for students to learn what these things are. Either way, we have Holiday Junction. And in the Holiday Junction, we do have crossing over. So we start with the same process we have before. Rec A, for example, will bind to the free overhang, invade, and we get displacement. So here's where the step is going to diverge. Instead of this strand not doing anything, when it gets displaced, it goes over and base pairs to the other partner. And that's where the crossing over is happening. Because now we've moved one piece of DNA from its original molecule to a different DNA molecule. And as we go through the process, we're going to see that red is now completely moved to the other side relative to where it started with. So as we fix this, we're creating a weird chimeric molecule. That's both things get changed in some way or another. And it's kind of hard to see it in this picture. So as we look at a different picture, you can see how the two strands of both DNA molecules become intertwined together. And this wouldn't happen without crossing over, because without crossing over, this red piece would not base pair to the blue piece at all. It would just be hanging there by itself. And this is showing you after the repairs already happened. So we've done all the fill-in and stuff. But the blue and red stay intertwined together, so when you fix this or to pull it apart, you gotta cut the DNA and you can get sort of these types of results at the end of the day. And what this is trying to show you is that change is possible. Up here, the capital A and the capital B were both on blue, which we call cis, right? After Holiday Junction, capital A and capital B are now not on the same chromosome anymore. So combination repair with crossing over is crossing over. So we do see that cis-trans switch as a possibility. I put together my own little drawing of things for fun, try to help illustrate this. I don't know, the Sharpies I used, the colors have kind of faded over time before I scanned the picture. So I apologize that yellow doesn't show up. That's yellow, and that's blue, and that's yellow. It's weird, it's kind of hard to see. But again, one side is crossing over, sorry, that side is crossing over, this side is without crossing over. So we heteroduplex over here versus Holiday Junction over there. But what I want you to focus in on is, without crossing over, the template strand is exactly the same at the end, at the beginning, without crossing over. But with crossing over, our template strand that's all yellow and red there is now got some blue parts in it because of the crossing over that happens. So that's what's important about the crossing over part, is both strands get changed. Whereas in the other way, only the fixed piece might be different, or will be different in some way or another. And then it's kind of hard to see, but you've got a piece of DNA here, or a strand of DNA, blue, and then yellow, and then blue again. So you're definitely getting this whole piece right here was from the other DNA molecule completely. Because it's red, yellow from the other piece of DNA. Okay, what you need to know is the basic steps. Definitely know some of the proteins, there's only two. They're at the very beginning. And then the names of the different structures that are formed during the crossing over mechanism versus the non-crossing over mechanism. I never asked you to draw this, but every once in a while I put a picture to see if you understand some concepts. What's the most important thing about this? Can't really tell on this slide, but what was the most important thing that was going on during this process? Anti-parallel. Very important thing to think about. Things have to go anti-parallel. this blue piece, this light blue piece, would never base pair to the pink piece because they're both going in the exact same direction. So they would be together, we wouldn't be anti-parallel. Always anti-parallel. This is just a summary of some of the things I've talked about already. We do have one other way to fix double-stranded DNA breaks. And we also have a fun little molecular tool that works with a similar mechanism as these repair mechanisms. And I guess wait until Friday to hear about that. Have anybody heard of a thing called CRISPR?